Hi everyone. For today, we will learn subtopic 2 in recombinant DNA technology. In 8.2 is about methods in gene cloning. Learning outcomes for 8.2 is 1. Overview using diagram to show the steps in gene cloning by using plasmid. 2. Describe the steps in gene cloning by using plasmid as the vector. 1. Isolation of gene. 2. Cleave or cut. 3. Insertion. 4. Transformation and amplification. 5. Screening, blue-white screening.
Recombinant DNA technology has gained paramount importance in the 21st century. In times when agriculture is faced with challenges such as drought, diseased crops, and poor yield, this technology has a vital role to play in developing better crops. Recombinant DNA technology has also helped produce medicines to prevent and cure diseases such as sickle cell anemia and cystic fibrosis. Recombinant DNA technology is a sophisticated process involving many steps. In order of sequence, these steps.
Here are the overview steps in gene cloning. 1. Isolation of gene. 2. Cleave or cut. 3. Insertion. 4. Transformation and amplification. 5. Screening, blue-white screening. The steps in gene cloning is 1. Isolation of gene. 2. Cleave or cut. 3. Insertion. 4. Transformation and amplification. 5. Screening. The first steps in gene cloning is isolation. We have 2 isolation, that is 1. Isolation of plasmid from bacteria that we will use it as vector, and 2. Isolation of target DNA or gene of interest from human cells, plant cell, or etc. Second steps is cleave or cut. Target DNA and plasmid are cut at palindromic sequence, that is, restriction site, with same restriction enzyme. Here is the example of DNA that cut by restriction enzyme, that is EcoR1, and produce complementary ends. When cutting with the restriction enzyme, the plasmid will open and it will disrupting the LAC-Z gene. For target DNA it will produce thousands of fragments with different sizes. The third steps is insertion. The DNA fragments are mixed with plasmids. Plasmid base pair forms hydrogen bonds with complementary target DNA fragment. DNA ligase is added. DNA ligase will join the DNA fragment by phosphodiester bond, forming recombinant DNA. But, not all plasmid will join with the target DNA. We have 2 possibilities, that is, 1. Non-recombinant plasmid. That is plasmid that rejoin without taking foreign DNA. 2. Recombinant plasmids. That is plasmid that rejoin with taking foreign DNA. The fourth steps is transformation and amplification. In this steps the host cells, that is bacteria, will take up recombinant DNA after being introduced to it. Bacteria and recombinant DNA are mixed in a medium containing calcium chloride. This calcium chloride will cause the bacterial cell wall to become permeable. But, not all host cells will take in the recombinant DNA. Only a small proportion of bacteria will be transformed. There will be 3 conditions, that is 1. Transformed with non-recombinant plasmid. 2. Transformed with recombinant plasmid. 3. Not transformed. Then, the host cells reproduce by binary fission, and it will amplify the target DNA. The last steps for gene cloning is screening. This steps is important because it will use to identify bacterial colonies that carry gene of interest or target DNA. This screening we called it as blue-white screening. A plasmid vector contains two genes. 1. AMPR gene, that encoding for resistance to antibiotic. 2. LAC-Z gene, encoding for enzyme beta-galactosidase. When cultured in medium containing ampicillin and X-gal, 1. Bacteria without plasmid fail to grow. Because this bacteria do not have AMPR gene and cause this bacteria not resistance to antibiotic. 2. Bacteria with plasmid live but with two conditions. 1. Contain non-recombinant plasmids. LAC-Z gene for this bacteria that encodes for beta-galactosidase is functioning. Bacteria can hydrolyze X-gal. So, they will become blue colonies in the medium. 2. Contain recombinant plasmids. LAC-Z gene for bacteria that encodes for beta-galactosidase is disrupted. Bacteria cannot hydrolyze X-gal. So, they will become white colonies in the medium. Now we can isolate and culture the white colonies to obtain bacteria with the inserted gene.
Blue-white cloning can make it much easier to select transformants that contain the insert of interest. With this technique, finding the right colonies is as easy as ignoring the blue colonies and focusing on the white ones. To perform blue-white cloning, you'll first need to make sure your competent cells are compatible with the technique. These cells are described as LAC-Z delta M15, meaning that they carry a mutation that deletes part of the beta-galactosidase, or LAC-Z gene. This results in a protein that only contains one of its necessary subunits. You'll also need a compatible plasmid, which contains a multiple cloning region within the coding sequence of the piece of LAC-Z that was deleted from the competent cells. This plasmid alone will restore LAC-Z activity, but successful ligation of your insert will disrupt the LAC-Z coding sequence, so the activity is not restored. Using these components, you can run your ligations and transformations as you typically would. When you're ready to plate the cells, make sure you use plates that are treated with the antibiotic you're using as a marker. For blue-white cloning, these plates should also be treated with
5-bromo-4-chloro-3-endolyl beta-D-galactopyranosidase.
That's a mouthful. You can just call it X-gal. X-gal is the key substrate component for making the colonies turn different colors. Once the cells are plated, incubate them overnight as normal. Now, it's time to check out your colonies. Since the plates were treated with an antibiotic, the colonies most likely are made up of cells that took up your plasmid containing the resistance gene. Otherwise, they wouldn't have grown. Take a closer look and notice that some of the colonies are blue while others are white. LAC-Z activity in the cells will turn them blue, so you can conclude that blue colonies do not contain your insert. The white colonies do not have restored LAC-Z, so these contain successfully ligated DNA. These are the ones you will want to use for your future experiments.